In this lecture, I will talk about mixed models, and in particular how it ties in with certain aspects of experimental design, such as blocking and nesting. So first I'll talk about some statistical considerations of study design. We already discussed some during the model selection lecture, but there are other important considerations, namely balance, replication, randomization, and blocking. Now, especially the last three tie in with the use of mixed models, which is why I've included them in this lecture specifically. Then I'll give a brief introduction to mixed models. You're probably already familiar with mixed models if you're watching this from Howard's lecture, but it's a complicated subject, so it doesn't hurt to hear from it from two different lectures, I think. So I'll give you my explanation of what a fixed and random effect is, and then I'll go into a little bit more depth on how you can interpret a random effect. And then I'll talk about the choice of using a random intercept or a random slope, or both, and why you would want to use one over the other. And then I'll show you how you can incorporate all of that in R. And then lastly, I'll talk about the most important subject of this particular lecture, which is nesting. So if you have a properly designed study, then you'll have maximal statistical power. So the ability to demonstrate an effect, whether you're doing it for hypothesis testing or just for inference, or even just for prediction, is dependent on a proper study design, on your sample size, balance if it's possible, which I'll explain in the next slide, and the choice of model. And also, if there's already a pilot study or there's previous research data on this subject, then it's possible to simulate the entire model, provided that you already have an idea of what kind of study design you're going to do, to get an idea of an appropriate sample size. Now, you're not only using proper study design to have a more powerful study, but also to increase the translational value of your research. So the first question you should always ask yourself is, can you generalize the conclusions to some larger population? So for example, people from the Netherlands included in a study versus people selected randomly from all over the world. That's a very different sample. And also, is the sample representative of the population of interest? So there are a lot of studies about health and human condition, and in almost all of these studies, we look at people that aren't actually healthy. We're looking at obese people, or we're looking at only athletes. And in this case, you can't really generalize to the general health of a normal human body. So depending on what kind of conclusions you want to draw, you have to select a different kind of sample. Now, besides the power of your study and its translational value, if there's actual flaws in your study design, that can also completely invalidate your conclusions. So you should always think carefully about whether your groups are comparable and whether samples were drawn in an unbiased manner. And this is also one of the reasons why we randomize. And whether your samples are true replicates or pseudo-replicates. So balance, randomization, and replication are the things I'm going to be talking about next. So for categorical comparisons, a study requires a sample of each condition, of course. But if possible, you should also try and aim for an equal sample size per category, and this is what's called balance. Now, if you don't have categories, there's no such thing of balance, but you can imagine something similar for a continuous variable. So if you're studying the effect of a continuous variable on the outcome, then yeah, you need at least 3 variables to estimate slope and intercept and have 1 degree of freedom left for the residuals. But ideally, you would also want the samples that you draw to be more or less equally distributed along the continuous variable. Now, balance is nice, but it's not required, and it's also not always possible. But if it is possible, you should try and aim for it because it increases the power of your study, and it also greatly increases the simplicity of analysis. Right, next we're going to talk about replication. So the required sample size depends on the variance, so the stochastic part of a process, the effect size, so how large are the true differences in the population, but also on the model that you're going to use to analyze the results. because if your model has more parameters, then you need more samples to estimate all of those parameters. So variance in effect size can be determined from a pilot study if it's available, or from previous research, or even just from expert knowledge. So for example, if you don't know how large the difference in the population are, you can still define a minimally relevant effect size within the context of your research. And then you can simulate what the appropriate power would be given that effect size. And of course, if you're an experienced biologist, maybe you're able to make an educated guess of the variance, even if you don't have data on it. So model complexity depends on what kind of comparison you want to make and what kind of distribution you think the outcome has, because some distributions have more parameters, and yeah, more parameters means you need more samples. And also whether you believe there to be potential confounding effects that you want to include, etc. So I've also written in Introduction to Biostatistics, paragraph 3.5, how you can determine a minimal sample size, which is of course not something that I would generally recommend, but yeah, if you're just doing a pilot study or there is no data available on how many samples you need because you can't estimate the variance, then yeah, it can be useful to start with a minimal sample size and then later progress to a complete study. So the required sample size always concerns true replicates. So be very careful when you think of sample size. It's not just the number of observations, but it's the number of independent observations. Now, pseudo-replicates don't increase your power, and they also don't help you meet the minimum number of samples. So yeah, I've given two examples here from chapter 2. True replication is when you have distinct experimental units, so it could be 5 different plants or 5 different patients, etc. Yeah, but in some study designs you want to repeatedly measure something like before and after, and that's fine, but it doesn't increase your sample size because those measurements are not independent. So yeah, it's always important that you think about how many true replicates there are in your study. So we've talked about balance, which is nice if possible, but not necessary and also often not possible. We've talked about replication, and that's a bit different. True replication is absolutely essential. So now let's talk about randomization. So randomization, also covered in 3.5, is the random allocation of treatments, locations, or even the order in which you process samples. Now, why would that be beneficial? For example, imagine you use a spin column to remove high abundance proteins in a proteomic study. Then these columns can easily cost in the hundreds of euros. We work with those here at my laboratory, and they're very expensive. So we try to reuse them as much as possible, and the manufacturer says that that's fine as long as you clean them properly, and you can use them up to 100 times. But even if the cleaning would be perfect and there would be zero residue of any previous samples, which is unlikely, then still the efficiency of the column slowly decreases, which is why we only use it 100 times maximum and not a million times or something. So eventually, it doesn't filter out the high abundance proteins as effectively anymore. Now, that also means that if you keep using the same column over time, then with each experiment, the efficiency is slightly lower. So in this example, let's say that you've already used a particular column 40 times and you have 25 samples to use, then the efficiency with which high abundance proteins were removed in the first sample will be very different than in the last sample. And the effect might be more pronounced or less pronounced depending on what kind of confounding effects there are in your study, but the effect is always there if you analyze samples in a structured manner. Now, if you randomize, then you scatter the order in which samples are organized. So if there's any systemic effect of doing something over time, which could even be as simple as the learning effect of doing the same experiment over and over, which reduces the measurement error, if you randomize the order in which you analyze your samples, then it doesn't have a structural effect on the outcome. So here's another concrete example. If you would have two groups that you want to compare, so 12 samples for the first and 12 or 13 samples for the next group. If you were to first analyze all the samples in one group, then on average, the measurement error for the first group will be different than the measurement error for the second group, because in this example, the efficiency of the column is reduced over time. And when you randomize, then the samples from A and the samples from B have no average difference, because the efficiency of the column was picked at different random times in the processing of samples. So that's what randomizing does. It removes potential bias. And this is an example where I know that the column efficiency reduces over time, and this is something we measure and we track. But often you don't know what causes bias, and it could be very subtle, and it could be the sum of many different things that change over time. So whenever you're in doubt, always randomize the order of treatment allocation. And not just treatment allocation, but also the order in which you run your experiments. Alright, we've covered balance, replication, and randomization. And now the last thing that is very closely related to the use of mixed models is the subject of blocking. So a block is a subset of the experimental material within which experimental units are expected to be homogeneous. So what that means is, if for example you have some experiment with several microarrays, because you have so many samples that they don't fit on a single array, then you can imagine that there are small differences in the manufacturing process and the cast or whatever that causes there to be minor differences between each microarray. And especially for something as sensitive as a DNA microarray, these differences can actually be picked up in the analysis. So in this case, one single microarray could be considered a homogeneous block. The idea is then simple. You randomize your treatments or conditions within blocks instead of over the entire experiment. So in the first example, you would randomize separately for each microarray. And let's say that you have some agricultural study where you have different plots of land, then you would want to randomize over each plot and not over all of the available land at once. And another example, let's say that there's a very large clinical trial involving multiple hospitals. Then you want to randomize the allocation of treatment, so placebo or the actual treatment, per hospital and not over the entire clinical trial. Because then it might happen that one hospital did not use one treatment or use very little of one treatment. And that's something we want to avoid because we also pursue balance. So there are also certain randomized designs that are especially appealing from a mathematical point of view, like the Latin square. But this is beyond the scope of this course. If you are interested in study design and clinical trials and stuff, then there's a book that I can really recommend to read more about this, which is not very mathematical, written by Wellham et al., called Statistical Methods in Biology. Right, so the reason to block is to reduce the bias and variance from measurement error or just differences that naturally occur. And it also allows for nested, hierarchical, or mixed models to be used to their full potential by estimating what the offset of each block is, so how it affects the outcome. This not only makes your life much easier during analysis, but you won't have to be concerned about any potential confounding effects of the blocks. And the way that we do it is by estimating a random effect in a mixed model. So that's what I'm going to show now. So I'll give a brief explanation of a mixed model, even though you've probably already heard this yesterday if you're watching this. But again, it's a difficult subject, so maybe my perspective can help you understand. So a mixed model is simply a model that has both fixed effects and random effects. And we include random effects to model dependency or structure or hierarchy in the data. And a block is one of those examples of structure. So let's go over the difference between fixed and random effects first. A fixed effect is a variable that has a constant effect, or something that can be measured without error, or something that is set, so fixed, by the scientist. A rule of thumb to recognize a fixed effect is that if measured twice, then the value should be the same. So these variables are assumed therefore to be measured without error. So an example is age, or a person's sex, or the type of treatment applied, or the dosage of a treatment. And the first two we can assume to be measured without error, and the last two we set ourselves when we design the study. So in practice, there may be measurement error. You think you applied a dose of 100 milligrams, but it was a half more or half less, but we assume it to be 0. And if you measure at a much later time, then age has changed, of course, but at the same time and location, these variables are constant. Now, a random effect is a variable that introduces dependence in the data. So, for example, let's say that you measure patients twice, before and after treatment. Or let's say that you have students from multiple schools which are included in your study. Or you're performing a meta-study which includes results from multiple original studies. Now, you can imagine that in each of these cases, the patients, the schools, or the studies are more closely, observations from the same patients, schools, or studies are more closely related to each other. And if measured twice, as a rule of thumb to recognize a random effect, these could be different. In another study, we would use different patients. In another study, we would use different schools, and we would use different original studies. But for a single patient, or for a single school, or for a single study, all the fixed effects would be constant. So the effects of these variables, random effects, are assumed to be 0 on average, with some unknown variance. And the goal of a mixed model is to estimate this variance and then subtract that from the overall effect on the outcome. So there's of course exceptions to the rule. It's just a rule of thumb. So let's say that you want to compare a specific set of patients. So for example, there's a clinical trial that went wrong, and you want to compare the results to a clinical trial that didn't go wrong to see what happened. Or you're comparing the performance of a specific set of schools to report on those schools, and not some more general idea of how schools perform. Or let's say that you're contrasting two specific studies rather than performing a meta-analysis where you're not interested in any specific studies, you're interested in the overall effect. So in these cases, the effect of patient, school, or study would be fixed because you're interested specifically in the observations that you have now. All right, let's go over the notation real quick. You don't have to memorize this, but I just want to show you because we also showed the notation of the t-test and the linear model, etc. So in a simple linear model, we would have some response variable y, and then we have a fixed part consisting of the intercept and the slope of x, and then we would have a random part, which we call epsilon instead of beta, and the random effect itself we call z, because x was already taken. And of course, we still have a random deviation epsilon. All right, if you then consider an example with multiple explanatory variables, the only thing that changes is the size of the fixed part. We could have multiple explanatory variables, but still we can just add a random part to account for the dependency. And just like the error term, which is epsilon does not have a fixed effect, which is why we call it a random effect. But it comes from a normal distribution, or at least that's the assumption. So this is how you write that. Epsilon is distributed normally with a mean of 0 and sigma squared epsilon. And epsilon, so epsilon sounds similar, but the U looking sign is also distributed normally, also with a mean of 0, but with a different variance, sigma squared epsilon. So that's what the subscripts mean. It just means that they each have their own normal distribution with their own variance. But again, this is just for completion. You don't have to memorize this notation. All right, so let's look at an example. Let's consider 12 patients in a clinical trial on chronic hypertension. So that's too high blood pressure. And we measure them once each week for 9 weeks in a row after we've begun some treatment, which hopefully reduces their blood pressure. And I simulate these data, so of course the blood pressure reduces. The point here is, if we look at each color, which is a different patient, then you can very clearly see that the results of one particular patient are correlated to each other. If you started out very high, then you also end up higher than the rest. And if you start out very low, then you end up lower than the rest. So that's the first observation to make. So, namely, each patient usually has a slightly different overall value. And what the random effect does is it estimates a normal distribution from which these patients were drawn, so that we can correct for each patient's random deviation. So yeah, with the LME4 package, you can do this very simply. You make a linear mixed model and use the function LMER. And then I say blood pressure is a function of the logarithm of time plus 1. So I chose the logarithm because this doesn't look linear to me, but you know, it looks like it has diminishing returns, which also makes sense because if the blood pressure would keep decreasing, then the patient would die. And I do the log plus 1 of time because yeah, time starts at 0 weeks since treatment, and the logarithm of 0 is undefined. So I say blood pressure is a logarithmic function of time, and I add a random effect, which is this part, for patient. So 1 means intercept, vertical bar means yeah, over, so patient is then the random effect. So this is the notation in this package. If we produce a summary, we can see what the mixed model does. First, it just shows a call, which is yeah, what we entered in R, and then it shows a convergence criterion. Now, if your model converges, then you won't get any error or warning messages, and then you don't need this criterion. But if you get an error or a warning, you know, about singularity or yeah, other problems, convergence not reached or something like that, then you should look at the convergence criterion, because what you're probably going to do is change your model. Maybe it's too complex, maybe you misspecified the random structure, and then the convergence criterion should have changed after that. If it doesn't, then there's just probably a different problem going on. So that's what you can use the convergence criterion for. Right, then we see the scaled residuals. Yeah, this is not something that you have to pay attention to because we can just use a QQ plot to look at the residuals. But yeah, as usual, the median should coincide with the mean for normally distributed errors. So if the errors are truly normally distributed, then the mean of the residuals and the median thus as well should be more or less 0. Well, this isn't far from 0, so this is fine. And another property of the normal distribution is that it's symmetric. Well, this is the first quartile and the third quartile, so that means the first 25% of the data and the first 75% of the data. And, you know, this is about as far from 0 as the other one is, so it's more or less symmetric. And, you know, the minimum and the maximum, you don't expect those to be symmetric because they're extreme values and they behave differently. But let's say that the maximum value would be like 100 times higher than the minimum value or something like that, then it's probably an outlier. Alright, then we get to the interesting part, because this is the part that makes it a mixed model, namely random and fixed effects. So in the random effects type, we see what we did. Namely, we have a random effect for a patient encoded as an intercept, here the 1, and the model has estimated a random variance of 17.5, and standard deviation is just the square root of that. And then it also says the residual variance is 0.8, with the square root of 0.8 is 0.9. So yeah, what does this mean? This means that, well, among other things, the variance of patients is apparently a lot higher than the residual variance. So let's see if the data agrees with that. That would mean that if we look at the logarithmic relationship, then the deviation from the logarithmic relationship, the residual, should be less than the difference between patients. Well, I think this is very clearly the case. The difference between patients is rather large. And, you know, some observations are up where you would expect them to go down, and this blue one is not really where you would expect it. That's the residual. And these differences are much more subtle than the difference between patients in this example. So this is not a general feature of mixed models, but yeah, if the random effect is larger than the residual, then this is what it looks like. Alright, and then the last part I don't have to explain because you already know about mixed models if you're watching this. So this is just the estimate and the effect of our transformed variable, the standard errors, and the t-value. Now you'll notice there's no p-value here, and that's because for mixed models, p-values are a bit more tricky to calculate. So if you really need them, well, you should probably contact us. And if you're using a GLMM, which we will discuss later, then you really shouldn't produce p-values, but you should instead try and bootstrap a confidence interval. But these are all things that you'll learn later in the course. Alright, so let's have a closer look at the random effect. We have 12 patients, and if we say RANF, random effect of LMM, which is the model I just made, then we can see the estimated offset of each patient. So what this means is that there was a normal distribution estimated with a variance of 17.5 or something. And then, yeah, each patient has been allocated to a particular position in this normal distribution. So, yeah, if you recall from the first figure, the brown patient was a lot more higher in blood pressure, and the green patient was a lot lower in blood pressure. And, you know, the yellow one was more or less in the center. So, let's compare that to the original data again. And then we can see what the random effect does. Namely, it estimates, all right, if we look at all these time points, then, yeah, how much up or down is every patient from the average linear effect. And yeah, it's a linear effect of a log transform variable, so it's a logarithmic effect, but it's still a linear model. Yeah, if you imagine a logarithmic line here, then what the random effect does is it says, how does each patient deviate from this line? So, what's really great about this is that imagine if you would include a fixed effect for patients. Then you would have to calculate 12 different effects of patients, right? You could put one in the intercept and then you have 11, but still that's 11 degrees of freedom. And now, all we did is estimate a variance, and then we're done. So this doesn't require a lot of degrees of freedom, so it's very efficient. Yeah, another thing is that this takes into account the difference across all time points. So it doesn't just look at a baseline difference or something. So sometimes you'll see studies correcting for baseline measurement, but I wouldn't recommend this. I would just recommend using a mixed model and including the baseline measurement. All right, so that was an example with a random intercept because it's easy to understand, but mixed models are actually really versatile. And by default, yeah, I would start with a random intercept because that is sufficient to model the dependency in the data. And also it's very efficient because all we need to do is estimate one variance. So a random intercept is like an offset for each category of the random effect. So in the examples before, it could be the offset that a patient has on the outcome or that a school has. You know, some schools are better than others. They might have different teachers, etc. And the difference between individual studies coming from different labs with different equipment, etc. But you can also use a random slope. So yeah, in this example from the book, which I'll also post online, it's not mandatory because it isn't finished, but yeah, if you want to have a look at it, you can read chapter 2. But the random slope is like a difference in fixed effect for each category of the random effect. So in this example, we're looking at the circumference of trees as they grow over the months. And, you know, a random intercept would be like thinking that every tree starts out at a different circumference and then starts growing in the same manner. Whereas a random slope would be to assume that every tree starts out at the same point when you start measuring, but, you know, they grow at a different rate. And then the differences in rate are accounted for by the random effect. The last effect is if you would do both a different starting point and they can have different progression. So this is the most versatile model, the most flexible model. But yeah, of course, more complex models require more samples. So in practice, you will very rarely use this because yeah, it requires very, very large sample size to be estimated effectively. All right, so yeah, that's why I gave an example with a random intercept and not one with the slope, because a mixed model with a random intercept is very efficient. You only need to estimate the variance of the random effect. And yeah, like I said before, if you would have used a fixed effect, you would need an estimate for each individual category. So it's way less efficient. Now, random slopes allow for very interesting and flexible models, and sometimes they are more correct from a theoretical standpoint than a random intercept, but they do cause more degrees of freedom. So even if you think you need a random slope, your sample size might not allow you to estimate one. So yeah, you need a large sample size, and remember from the earlier part of this lecture, when I say that, I mean you need more true replicates. So that would mean more patients and not more time points of the same patients. Alright, so in R, I already showed one mixed model, but actually almost all mixed models are easy to implement in R. It's a complicated subject, and conceptually a mixed model is a lot harder to understand. The estimation is even more complex. It's called restricted maximum likelihood if you want to have a look. But yeah, in R it's very simple to implement with this really great package LME4. So if you want to have a random intercept, you just say, all right, I have a linear mixed model, and then you say LMER. You have to load this library for that. And then you say response variable. Then you do one or several explanatory variables. And, you know, as normal, you can include interactions if you want. And then at the end, you say plus, and then you include the random intercept. So if you say 1 over the random effect, then that means intercept. If you say 0 plus x, 0 means not an intercept, and then plus x means I want a random slope for variable x. So you can do it for different explanatory variables if you would have several ones. And lastly, you know, it's possible to do both. So in this case, you would say x, because by default, there's always an intercept included. So yeah, if you would type 1 plus x, that's the same as the previous one. Right, so again, this isn't part of this lecture yet, but I just want to show you how easy it is to further add to the complexity of this model. Let's say that you want to use a mixed model, and your data cannot be assumed to be normally distributed conditional on the explanatory variable. So let's say that you're working with counts and repeated measurements. Then you could use a Poisson GLMM. Or let's say that you're working with ratios, like the number of people that got a disease and the number of people that didn't under various conditions. Then you could use a binomial GLMM. And it works almost the same. It's the same library. All you have to do is call the function GLMER, so generalized linear mixed effects regression. And, you know, after you've specified your model of interest, which could be any of these, or you could add more explanatory effects, then you add an argument family, and then you say the underlying distribution that you think the response variable should have. So GLMMs, very easy to implement, but a lot harder to work with. So if you need one, I would definitely recommend that you consult with a statistician. And, you know, if you're watching this in the context of the course, of course, you can always come to us with questions. Right, so a mixed model can not only account for these single dependencies that I mentioned, but you can have way more complex structures or hierarchy in your data. So let's say that you have a very large clinical trial involving repeated measurements of multiple patients from different hospitals and across different countries across the world. Because like I said before, if you want to generalize the results to the whole human population, you can't use one country. So we have multiple countries and multiple hospitals per country. And then it could also even be a repeated measurement study, like before and after measurements, or the 9 measurements of blood pressure that I just showed. And this is a bit more complex because there are 3 random effects: patients, hospital, and countries. And they're nested because a hospital is within a certain country and a patient is within a certain hospital. So there's nesting going on here, or hierarchy in the data. Another example, let's say that you have some agricultural study on environmentally friendly insecticides, and you want to include the results from multiple farms, each of which consists of multiple plots of land. Then, you know, a plot of land is a random effect, but plots of land are nested within the farm that they belong to. They can't suddenly switch farms unless they would be sold or something, but at a given point in time, then they belong to a particular farm. So you would have the effect of plot nested in the effect of farm. So yeah, one more example. Let's say that you want to develop some tests for properly standardizing high school exams in the European Union, for example. And you want to compare two different tests and see which one yields better overall grades. Then yeah, you could use students from multiple classes in each school, and you could use multiple schools from each country. And if you want to talk about the EU, you can't just do it in the Netherlands. Then you have to include multiple countries from the European Union. Now, yeah, one quick sidestep. Of course, you can't randomly draw countries from the European Union because you you have to agree to participate in the study. But for the purpose of estimation, you can assume that if you have a reasonable number of countries from the Europe, that you have a random draw of all the potential members of the European Union, like future, past, whatever. Yeah, you can treat it as a random effect, even though in reality there is a fixed number of schools in a fixed number of countries. But if there are enough of them and if they behave like a random draw, then you can consider the random effect. And also it allows you to account for this dependency, because even if there is a fixed number of schools, then still the students are nested in the classes, which are nested in the schools, which are nested in the country. So even if the rule of thumb doesn't work, you can still think of a mixed model in terms of dependency or hierarchy in your data. All right, so let's see how you would encode this in R if you had nesting. So again, conceptually, this is a more complicated problem, but again, using this package that everybody loves, it's very easy to implement in R. All you have to do is have your regular linear mixed model. So let's say the first example with a clinical trial across countries and hospitals and repeated measures of patients. Then we'd have some clinical outcome as a function of some treatment. And then we have this very large random effect, which says we have repeated measurements of patients nested in multiple hospitals, nested in multiple countries. So that's the notation. There's also another notation, but yeah, I think this one is the simplest one, so this is the one I use. Then, in the other example with yeah, standardizing an exam in the European Union, let's say we want to find the one that gives the best average grades. Well, the great thing about averages is that you would expect them to be normally distributed. So that makes sense to use LMER in this case. Yeah, and we have two different exam types that we want to compare. And we have students nested in classes, nested in school, nested in the country they come from. So at this point, you're probably wondering, okay, if students are in classes, why aren't students over here at the end? /student. And the reason is, if we don't have repeated measurements of students, then that's not a random effect. That's just one true replicate. So that's this note at the bottom. Only include random effects if there are random effects. So you need repeated measurements of it. So in the first example, we had multiple measurements of patients in multiple hospitals across multiple countries. But in the second example, we look at each student once performing exam number 1 or exam number 2. So we can randomly distribute the exams and then perform it all at the same time. And the students are then nested in their class, in their school, in their country. But for each student, there's only one observation. They did exam 1 or they did exam 2. So in that case, there's no random effect. So in general, for nesting, you would have a linear mixed effects regression as of a response distributed as some explanatory variable, or again, this is just for simplicity, but you could have multiple explanatory variables, x plus x2 plus x3, or you could do x times x2 if you have an interaction. And at the end, you have plus some random effect between brackets. Yeah, and in this case, a random intercept. And then if you have nesting, you first enter the superclass, so the highest part in the hierarchy, which could be a country in the examples. And then you do a slash, and then you do a random effect for the subclass. So that could be the hospitals in the country or the schools in the country. And if you have further nesting, just continue this hierarchy with slashes. All right, So that is all I have to say for this lecture. But I want to give you some examples because if you're watching this in the context of the advanced statistics course in 2020, then one of the things that you have to do is simulate data for a mixed effects model for the graded assignment. So for this purpose, I think it's a good exercise for yourself to try and look at each of these examples and see if you can identify what would be the fixed effects and what would be the random effects in each of these studies. Now, these aren't entire study descriptions because I just wanted them to fit on one slide, but just like for simulating data, you can come up with your own fixed effects or your own random effects that you might want to include. But based on these sentences, I already imply that there either is or there isn't some random effect. You don't have to hand this in. This is just to test your own knowledge. And if people like this and want to see the answers, then I can post some example answers on Brightspace later. All right, so in summary, we discussed some consideration of experimental design, namely replications, which is the number of true replicates. Then we talked about randomization to avoid confounding effects. Then we talked about blocking for estimating a confounding effect if it's there. We also mentioned balance, but I didn't even write it down because it's not a requirement. It's not even always possible. But if it's possible, you might want to try and achieve balance because it simplifies the study and it also adds to its power. But I think the key message of this lecture is that prevention is superior to cure. So you want to try and keep your study as simple as possible unless the research question demands it. There could be repeated measures that are required. If you want to see if in certain patients their blood pressure decreases, then, the best type of study would also include what their blood pressure was first and what it is after. So some research questions demand repeated measures or dependency in the data. Like if there simply aren't enough students to randomly draw from random universities, why not just include whole classes from different universities? So that requires dependency. Or you could have block dependency, like if you have to distribute the exams, like exam A or exam B, or you have to distribute a placebo or a real treatment, then you should randomize that per block. So not over the entire study, which may have nesting and random effects and whatnot, but per block. So that could be the microarray we talked about, or that could be per class of students, or it could be per hospital of patients, etc. So in each of these cases, if there is dependency in your data, you need a mixed model. And the last thing I want to say is pseudo-replication never helps with power, and it never helps you achieve your required sample size, but it can reduce, without a D, the measurement error by averaging over pseudo-replicates. But you don't have to average over pseudo-replicates because we just discussed this really cool mixed model, which can handle data with both true replicates and pseudo-replicates, just by including a random effect for whatever you pseudo-replicated. So that would be a lot more efficient than just averaging over the pseudo-replication, and it might end up giving you better estimates. And that's everything I want to say for this lecture. And I tried to keep it short because the only thing I really wanted to explain is the effect of nesting and how it might affect your study design and the other considerations that tie into your study design and what they have to do with mixed models. But if there's any questions about this, don't hesitate to ask. You can post them on Brightspace, or if the course is already over, you can post them below this video or something, and I'll try to answer them. Thank you for watching.